The process of meiosis involves two rounds of cell division. These are called meiosis 1 and meiosis 2. Prophase 1, metaphase 1, anaphase 1, and telophase 1, these are the steps of meiosis 1. Prophase 2, metaphase 2, anaphase 2, and telophase 2, that's meiosis 2. The purpose of meiosis 1 is to create haploid cells. That is, we're going to cut the number of chromosomes in these cells in half. Here I'm drawing cells with 2n equals 2 chromosomes, diploid cells with 2 chromosomes total. That means that the haploid cells at the end are going to have 1 chromosome each. In prophase 1, a phenomenon called crossing over occurs, where homologous chromosomes exchange bits of DNA. This means that some of the genes on your father's chromosome gets onto one of your mother's chromatids, and vice versa. This increases genetic diversity. In metaphase 1, we have random assortment of chromosomes, meaning that it's entirely random which daughter cell is going to receive which parental chromosome. Thanks to the genetic recombination of crossing over, each daughter cell at the end of this process will be genetically unique and distinct from the other daughter cells. In anaphase 1, we are separating homologous chromosomes, so we are cutting the total number of chromosomes in half. However, in anaphase 2, we are separating the sister chromatids on each chromosome that remains. So we are going to break the sister chromatids apart from each other and make them into daughter chromosomes. We'll end up with 4 genetically unique haploid cells. In order to observe the genetic uniqueness of each daughter cell at the end, I'm going to label some of the genes which are being carried by the parental chromosomes. On the father's chromosome, which I have colored in green, I'm going to give him genes for tallness and baldness. On the mother's chromosome, I'm going to place genes for shortness and hairiness, so a short, hairy chromosome. Now, during crossing over, each chromatid is going to exchange bits of that DNA, which means that there's going to be new combinations of these genes on each chromatid. At the end, we're going to end up with a parental chromosome that has the tall, bald genes, another one that has short, hairy genes, another one that has tall, hairy genes, and the last one will have short, hairy genes, just as the maternal chromosome would. After meiosis 1 and meiosis 2 have been completed, these sister chromatids, which are no longer genetically identical to each other, are each going to end up in separate daughter cells, meaning that we will have a sperm or an egg cell with the traits for being tall and hairy, another one with short and hairy, another one with short and bald, and the last one that is tall and bald. 4 genetically unique daughter cells at the end, even though we only had 2 chromosomes to start with.